have heard of genes that act selfishly, organisms that adapt in order to survive, ecosystems that behave as cohesive units. Biologists often talk about these entities as though they were purposeful, exhibiting agency, directionality, or function. Should they? That's the big question that brings together the experimentalists, theoreticians, and philosophers who are involved in the Biological Purpose Project. This is the Purpose Podcast, where we will sample different interpretations and applications of agency, directionality, and function in biology. Welcome to Episode 1. I'm Siddhant. Every cell in your body and mine began from a single cell. A single fertilized human egg divides into 2 and 4 and 8 and so on. Given the right kind of chemical signals and time, the cells form patterns, digits, organs, tissues, and eventually an entire interconnected system that we call our body. The guest of our first episode, Ramray Bhat, is interested in how groups of cells in our body organize themselves. But the cells he studies are rule breakers. At some point during development, most of the cells in our body lose their ability to divide, and they get down to the business of keeping us alive. One of the most important rules cells in our body have to follow is to keep themselves from making more cells. Sometimes, a cell will break that rule. It will become cancerous, develop into a tumor, and start spreading to other parts of the body. That's what cancers are, uncontrolled division of cells. And like our bodies,
all cancers typically arise from a single cell.
That's Ramray. He's an associate professor at the Indian Institute of Science in Bangalore, India. It takes a lot of mutations, what Ramray calls insults, for a cell to become cancerous.
And many of these insults sort of come in one by one and accumulate, and the right combination of these will ultimately decide whether a single cell becomes a tumor or not.
A cell could have potentially cancerous mutations, but it is only, as Ramray says, when a cell has the right combination of mutations that it becomes cancerous. And even then, many tumors are arrested in their spread. Ramray began his scientific career as a medical trainee at the University of Calcutta. It was there that he first learned just how riddled our bodies are with benign tumors.
When post-mortem is conducted on road accident victims or any accident victims, what one sees is that often when the body is opened up, you see that our body is full of tumors, but these are all benign. They all are present. Maybe they've grown a bit and then they've stalled. There's a lot that goes on, basically, that determines, even if one has mutations, whether it becomes cancer, whether it spreads, whether it grows, whether it goes to another organ, and so on.
So why are some cancers more successful at spreading, and why do they only spread to certain parts of the body? Over the years, researchers have investigated what is known as the tumor microenvironment, which is basically the cocktail of chemicals and normal cells of our body with which a cancer cell interacts. For some cancers, like breast cancer, this is relatively well understood.
But for instance, what determines when a breast cancer sort of starts migrating into blood vessels? How does it migrate in? What happens to the blood vessel cells when these migrations occur? Some of these things are not still understood. Again, once it gets into the blood, why is it that it goes to certain organs and not to some certain other organs? Is again something that is reasonably well known for some cancers, not so well known for some other cancers. So these are things that are still being investigated.
These are the questions that Ramray is interested in. That process that he was describing is called metastasis. That's when a cancer cell leaves the organ where it developed, enters the bloodstream, and successfully establishes itself in a new organ. Ramray compares the spreading cancer to a seed, and any new potential organ to where it can spread, he compares to a soil. The seed and soil metaphor, something cancer biologists have been using for over a century, and for good reason.
When cancers spread, they do so not just because of their potent abilities to spread, but also because of their surroundings, some of which can be hostile to their spreading, but some of which can also be encouraging And that defines why some cancers tend to spread to some areas in the body and not to other parts of the body. Muscles, for example, tend to be very poor areas for cancers to latch onto and spread, whereas bone marrows, liver, lung tend to be very fertile grounds for cancers to spread to.
And over the last decade or so, scientists who study cancer have begun to view metastasis a little differently. It became apparent to them that cancer cells sometimes work together.
There has been a big shift that has occurred since the 2007-8 or so, or maybe a little later, 2011-10 onwards, where there were some landmark findings that cancer spreads better when in blood it is able to be present as multicellular masses rather than single cells.
The shift in paradigm has been spurred by studies by multiple groups on different kinds of cancer.
Breast, pancreatic, ovarian, and so on, where now it is understood that, more than single cells, it's the clustered cancer cells that seem to mediate the spread.
It is almost as though they are stronger together. Ramray is on a quest to understand just why that's the case. Extending that seed and soil metaphor, he asks what is it that makes collectives better seeds than individual cells? And to begin answering this question, Ramray reflects on his years as a PhD student at New York Medical College.
At that time, we were working on a problem of how sort of digits form, digits meaning fingers, and how are they sized and spaced similar to each other, how do they come about? And we used a chicken vertebrate model, which is a classical model of developmental biology, to understand how limb development takes place and how limb pattern formation occurs within this paradigm.
A cell within a developing embryo, just like a cancer cell, experiences a complex cocktail of chemical signals which will determine its fate, whether it will become a skin cell, an immune cell, a neuron, and these signals will ultimately shut down the ability of the cell to divide. So if development entails an orderly division of cells, a formation of patterns, cancer represents its flip side, a deformation, a breaking of patterns. At least, that's what Ramray originally thought.
But in doing so, I sort of started digging deeper philosophically and started asking the question of when one therefore loses pattern, is it loss of pattern or gain of a new kind of pattern, a new set of principles that come in when one gets cancer?
So Ramray now believes that clusters of migrating cancer cells may have properties that allow them to work together, increasing their chances of successfully metastasizing to a new organ. He looks at these clusters as cohesive units.
When we talk about an organ or a tissue in our body which is functioning properly, it's because everything is cooperating with each other. The blood vessels are there to provide nutrition, the cells are there to perform a function, the connective tissue is there to give rise to a scaffolding. So all the cells are interacting with each other in a harmonious fashion. And cancer is thought to be the destruction of this harmony, a disharmonious sort of outcome. But then an emergent understanding in the field is it is not just the destruction of that harmony. Perhaps, as I said, new harmonious relationships come together. And so from thinking about cancer as the destruction of a collective back to singular entities, now cancer is thought to be the formation of new collectives, the formation of new organizations.
In describing the behavior of these clusters, Ramray refers to them as agents. A central question that people within the Biological Purpose Project often debate is what exactly that means. So far, they haven't come to a consensus. While admitting that he himself goes back and forth on his own definition of what an agent is, and while allowing for disagreement, Ramray says,
for me, anything that sort of is able to work to change its environment, but is also evolvable, is something that I would tend to call an agent. Now, one can think of it as a virus also is evolvable and changes an environment, and so is a virus an agent, perhaps. And I would be comfortable with that, but somebody else won't be comfortable.
Does that mean Ramray thinks tumors are agents?
They are able to sort of take different shapes, sizes, go from unicellular to multicellular, multicellular to unicellular behaviors. And so I would definitely be comfortable in ascribing agency to tumors.
An expanded perspective from the individual cancer cell to the collective has led him to look for properties that make them special, something that would explain why they become so much better at migrating and successfully metastasizing. The philosophical approach helped him broaden his understanding of cancer and take those questions to the bench, where he is studying cancer under laboratory conditions. There, he's trying to recreate not just the chemical environment, but the physical terrain through which a cluster of cancer cells would migrate. Ramray and his group are
trying to recreate aspects of the anatomy which we would find in our body, but under more controlled, imaging tractable, observation tractable platforms, such as on microscopes.
What he's trying to make are essentially lab-made three-dimensional structures that recreate the natural shape of organs and tumors. One big advantage of these is Ramray can observe the spread of cancer as if it was happening in the human body or close enough. Within this setup, his group is trying to
study these collectives of cancer cells and as opposed to single cancer cells, and try to identify whether there are special properties that come about at that collective stage.
In other words, what makes them so successful when they work together? You may or may not want to think about tumors as living entities. Cancer, even when treatable, is a life-altering diagnosis that affects millions across the world. But for scientists looking for cures, getting the right answer is often a matter of finding the right questions. And that's where a little bit of philosophy can help. If Ramray can unravel the secrets of the harmonious relationships between cancer cells in a cluster, he may well figure out how to break that harmony. Ramray's approach is helping him ask the questions he needs. Combined with cutting-edge experimental methods, they may eventually help him get to the roots of what makes collectives of cancer cells so special. That's all from episode 1 of the Purpose Podcast. The Purpose Podcast is brought to you by the Biological Purpose Project. To learn more about the Biological Purpose Project, you can visit biologicalpurpose.org. Thank you for listening.